So I'm going to talk about reducing label data needs. And in particular, I'm going to focus on microscopic and medical images with a specific scientific question in mind. So before we start, I just want to talk about the teams. So this is a collaborative effort. There are 3 teams involved. The scientists here are from PSI, specifically the group that does X-ray tomography. The people are Hector Valerdo and Anne Bonin. There's SDSC, specifically RK and Fernando is involved. And there is the Computer Vision Lab, which I'm a part of from ETH Zurich. The project itself has 2 different tracts. One of them is the scientific tract, and the other one is the engineering tract. In the scientific tract, the idea is to be able to accomplish macro and microstructure analysis of cardiac tissue. And this tract is being led by TOMCAT at PSI. While in the engineering tract, we're aiming to create machine learning algorithms, specifically for image segmentation, that can use low number of labeled examples. So I'm going to talk about both of these tracts, but most of the time I'm going to spend on the scientific question, which I believe was the main reason for the call at SDSC and better aligns with the goal. We want to investigate cardiovascular diseases. In particular, we want to investigate remodeling cardiovascular diseases. So how many of you have ever heard of what remodeling cardiovascular disease is? Great, so we're on the same track. I was exactly there about a year ago. So something happens to your heart. It's an infarct, it's a disease, maybe it's just like prolonged hypertension. Your cardiac tissue starts responding to this. As it responds, its structure changes, so your heart changes. It's a short-term solution with bad long-term effects. What happens, for example, is fibrosis, so your cardiac tissue stiffens, so it's no longer able to beat properly. Your ventricles get larger, your atrium gets larger, the shape and morphology of the heart changes. You have constant immune response because the scar tissue is forming and then it's broken apart and then it's forming again. And this is problematic. Because why? Because most of the time, the small incidences don't kill you. What kills you is the remodeling of the heart. So it's crucial to be able to understand this and to understand really how it works, why it works, and its effect. We have to take a multi-scale approach. We have to investigate at macroscopic scale, at microscopic scale, and really integrate the microorganization in the macro scale, in the tissue, integrate the tissue information in the organ scale. Now, there are various imaging techniques available to do. First part, which is actually used in the clinics, is to use medical images. Ultrasound, MRI, CT, PET, SPECT. And these images are amazing because they're used to diagnose these problems, but they're macro scale. We also have at our disposal histology analysis. For example, in the case where there's a transplant rejection, we can actually look at the heart outside and really investigate what happened at the microscopic scale with histology analysis. Now, although it looks like we have a lot of tools in our hand, it's not complete. Within this flora, there is actually space for more imaging techniques, and one of them is the X-ray phase contrast imaging, which the PSI group is actually an expert on. So to be able to motivate the need for this technique, let me go through what we currently have. We have, for example, ultrasounds. I'm sure maybe some of you actually went to the hospital and went through echocardiography. The ultrasound is great because it actually images in real time the whole heart and provides us functional information. The down part of it is only gives us macro scale information. The resolution is quite low. It's on the order of millimeters, if not more. Then we have MRI, a fantastic imaging technique that actually gives extremely high soft tissue contrast. We can image the entire heart. We can actually get functional information from it. We can get information about the alignment of fibers and so on. The issue is, if you want to get a good image, you need long acquisition times, and the resolution is still low. We cannot actually extract microscopic information and integrate to macroscopic models. We have CT, well, X-ray, multiple X-ray. It gives much better resolution than MRI and ultrasound for sure. It gives us the image of the whole heart, but the issue is it has very low tissue contrast, acquisition times can be long, and it's still at a macro scale. We can go down to, for example, 500 microns, but we cannot go down to 0.500 microns. Now, these are macro scale computation imaging. Obviously, there's traditional histology. So we can actually, take a heart, a biopsy from a heart, cut it, put it on slides, stain it, and image it. We get very high resolution. We get really good contrast because actually we stain for the things that we're looking at. But the field of view is limited, it's time consuming, and those of you who've ever actually worked with histology slices know that it's really, really kind of destructing the tissue. If you want to do 3D analysis with histopathology, with histology, you need to take into account the tearing, shearing, and all these artifacts, which becomes really, really difficult to do 3D analysis. The X-ray phase contrast imaging is a 3D, non-destructive method that you don't have to use staining. It's high resolution, it's time efficient, and you can get images at high field of view. The idea is actually quite simple. We simply have a very powerful beam here that's coming from the PSI, the Swiss light source. We have our tissue right in front of a detector and shine X-ray beams. They're quite powerful. This actually gives us very good spatial resolution. Now, the easy way of doing it is to use attenuation correction, attenuation contrast imaging, which would give us an image like this. So probably you see the shape of the heart, it's not very clear. The novelty of this imaging technique is if you actually shine coherent beams, phase coherent beams, then you can use something else. You can actually take the detector a little further away, again shine the beam. Now, instead of looking the number of photons that reach the detector, you look at the differences in the phases of the photons that reach the detector. In the end, you get really high resolution space and really good soft tissue contrast. Now, with such an imaging technique at our disposal, the goal here is, okay, let's use it to investigate what happens in remodeling cardiovascular diseases, specifically what happens to the tissue. In this project, we actually are looking for collagen quantification and assessment. So for that, let me give you a brief overview of what happens, why we're actually interested in collagen. So this is the heart, a very model, healthy heart. And if you look at a certain area right here in the muscle, you'll see that the cardiac muscle is formed of the actual cells that do the contraction bit, and the extracellular matrix. Now, the extracellular matrix has something called collagen fibers. These collagen fibers are quite important because they actually, form a nice scaffold structure so that the muscles, tissues, and muscle cells don't tear, don't stretch too much, don't contract too much. It basically gives a rough structure to it. What happens when actually something bad happens is that, well, let's say at this point, something happened to the heart, for example, a disease, for example, a cardiac arrest, an infarction. You have cell death. So most of these cells start dying, and you basically have leukocyte infiltration. The extracellular matrix that actually supports the cardiac muscle deteriorates. The response of the heart to this is to say, I have to solve this. So it actually, activates these fibroblasts, which basically secretes collagen in a sense. So it makes the tissue, the extracellular matrix, much stronger. Why? Because that's how actually scar wound happens. You basically, wound and then scar tissue forms. Scar tissue formation in the heart basically means fibroblasts synthesize extracellular matrix. Now the issue is the scar formation happens and remodeling starts. It's a short-term solution, and if overdone, leads to long-term bad effects. In the end, what happens is for example, the muscle cells, they died, and instead, the heart simply replaces with more collagen. What that means is the heart actually gets stiffened, and it no longer beats. And that leads to longer-term bad effects. So we want to basically investigate how and why this happens. Now, the link from the problems in the heart to the remodeling is well established. There is no question about that. What is unknown is actually the quantification. So how much collagen is formed? Because if we can have a good idea of how much collagen is formed, then maybe we can intervene at the right time. And furthermore, it's really unknown the association of the amount of collagen with the severity of the disease. So with this question in mind, our collaborators at PSI built a large imaging protocol. The idea is quite simple. We take biopsies from rat hearts in 3 different locations. We basically fix these samples, place them in this red tube right here, and put the red tube right in front of a camera. Now, the camera, right here, at the other end, is connected to the Swiss light source, which actually shines this coherent, powerful X-ray beam. And then we have 2 cameras, an HR microscope, a high-resolution microscope, and a low-resolution microscope. This guy here takes images at a very high spatial resolution. This guy here is at a low resolution, but has a much larger field of view. In the end, what it gives is it gives us images like this. So this is not just one image, actually. This is a 3D volume. And these are the 3 orthogonal views in that volume. Now, you have lots of structure here. I also put segmentations of these structures here. What we have here is the yellow parts are the actual cells. The blue areas, the small blue ones here, they're the collagen fibers, and we want to quantify how much collagen there is in the tissue. And the other parts, the rest, are basically background. Here, for example, I have 1 image from a healthy rat and 1 image from a hypertensive rat. And you see the amount of collagen is much higher. Now, this is a segmentation problem, so we approach it with a segmentation algorithm. We created a data set of endomyocardial biopsies of about 2 × 2 × 4 millimeter cube. So seems small, but we image them at 0.65 micron voxel resolution, so quite large images. We used 8 different rats, 2 of them as 3 months old, 2 of them at 12 months old, control rats, and 4 age-matched, spontaneously hypertensive rats with high amounts of collagen in the tissue. So we fixed the samples with 4% paraformaldehyde and placed them in the tubes and got the images. And we reconstructed images with and without Paganini reconstruction to get both the attenuation as well as the phase information. So the images were divided in subvolumes because it's quite difficult to feed all that volume into the computer, about 300 micron cubes. And we basically segmented the collagen end cells in 10 such subvolumes using elastic, a semi-automatic algorithm. We end up with such images. So these are the images without Paganini, with Paganini reconstructions. Segmentations of the collagen tissue, segmentation of the background, segmentation of the cells. And this would be the 3D reconstruction of the collagen tissue. So it looks quite nice. No? But notice that 10 subvolumes is a tiny bit of the entire data set. And it took roughly 10 hours of user interaction to segment them. So this is actually not possible to analyze the entire data set with this approach. So we need to do something. So what we did was simple. Take the images that are already segmented, use a very simple unit, train it, a segmentation convolutional neural network, train it with 8 subvolumes, 2 of them are used for validation, and we tested on 2 different subvolumes. And once we were actually satisfied with the model, we applied the unit, the trained unit, on the rest of the data set. And we first wanted to understand whether this segmentation can be done with a neural network. And then we did the analysis of the ratio of collagen versus cells for the scientific questions. So this is I'm actually showing the worst result. This is an orthogonal slice, again, the 3 different views. This would be the segmentation that the algorithm was able to extract, and this is the segmentation that we had in the ground truth. So overall, it's not a bad segmentation. It's difficult to analyze or understand it from those images visually. Here are the numbers. So as engineers we said, first of all, maybe we can actually solve it with a very simple algorithm. Maybe machine learning was not necessary. So we applied the Gaussian mixture model segmentation, and we saw that actually it's able to do something, especially when we talk about the dice score, so overlap between ground truth segmentation and the network's predict segmentation. We had some like 66% of agreement. When we integrated network, the agreement went up to 83% almost. What was crucial, though, is actually the ratio or the correlation coefficient between the ratio of collagen versus cells. So we have the ground truth, and we computed number of collagen pixels versus number of cell pixels, because that's really the important thing that we're after this in the scientific question. So we got them in the ground truth, and we got them in the predicted segmentations, and we simply computed the Pearson correlation coefficient. So the simple model, it's doing okay with the dice, but when it actually comes to the scientifically interesting question, the measurement, it's doing quite bad. Actually, the neural network that was learned from the algorithm, that was learned from the labeled data, was able to perform much, much better. So in the end, we applied it to the entire thing, and the first results will be presented, I think, in June 3rd or June 4th in the Functional Imaging and Modeling of the Heart conference in Bordeaux. What we did was we actually analyzed the entire set of images, and we observed two different facts. One of them is there's a difference, like as the rats get older, the amount of collagen in the heart increases, which was actually observed in a previous publication by Legris et al. And furthermore, we also noticed that in some parts of the heart, there is actually a difference between the control rats and the rats that were hypertensive. So hypertensive, as expected, really led to higher amounts of collagen. Obviously, a much larger scale analysis is needed, but within the context of the project that STST funds, this is basically the initial demonstrations that high throughput, large sample analysis is possible using machine learning algorithm, and that was the first step that we wanted to make. Now, that was the scientific tract. I'm going to quickly talk about the engineering tract as well. So in this question, remember this slide? It took more than 10 hours of user interaction to do this. The thing is, this tells us if you're able to put this amount of time, great. And most of the applications, you will not be able to put that amount of time. Furthermore, this is rather okay segmentation problem that could be solved with a semi-automatic tool. But most segmentation problems are not like that, so it would take a lot longer. So cutting the labeling cost is actually essential. And it's not just essential for the microscopic images, it's essential for medical images, it's essential for any type of images. If I want to, for example, segment different organs in volumes of CTs, the labeling part is extremely crucial, yet extremely laborious. So one of the goals of this project, the engineering goal, was to reduce this need. And for that aspect, we basically did a simple analysis. We took the data set from the PSI, the micro-CT data set that showed the collagens. And we progressively reduced the amount of training data. And we trained units to really see what is the effect of the number of examples to the accuracy of the prediction. So it started with 80% of the data trained and went down to 0.006% of the data trained. So 0.03 roughly corresponds to one slide being fully segmented, and afterwards it corresponds to segmenting many slides, but with only clicks rather than full segmentations. So much, much, much fewer samples. At this point, we actually only have 3 slices. Each slice has 45 clicks. That's it. So 45 clicks is about 13 slices, and this can be done in 1.5 minutes, so roughly 5 minutes of segmentation time. So first of all, the unit is great. It's like neural networks actually do work. We could get really high segmentation accuracy even with only one labeled slice. Reducing the label further down, we see that actually the accuracy is decreasing. So what we want is, we want to be able to label this much, but get this much accuracy. So we tackle this problem, we're attacking this problem in 3 different directions. First is basically trying to use clever ideas with data augmentation. Specifically, we're considering semi-supervised, task-driven data augmentation, which is the second output from this project, which will be presented also in June in Information Processing Medical Imaging Conference. So what you see here is actually cardiac MRI, and this is the ground truth. Normally, what we want is we want to be able to segment this with one labeled examples. And through different ways, you can actually get quite good accuracy. And what we figured out is actually if you optimize the data augmentation process, you could get quite a good segmentation using only one labeled image. How to do it? We basically really optimize or augment our training data set with an augmentation that's somehow optimized within the training process. The second track is actually weak learning, or learning based on editing with scribbles. On one side, we want to be able to take the scribbles that are made by the user, for example, the clicks, and we want to be able to propagate them through the entire image in a clever way, so that we can actually retrain the algorithm with that propagation. And that leads to really good segmentation algorithms, even with one or two labeled examples. Another one is, most of the time, you do a segmentation. Segmentation is not perfect, you want to edit it. So we're investigating algorithms that are optimized to improve the segmentation with the clicks. So really integrating neural networks, integrating machine learning in the editing process. And the very last, now coming back to the problem, we're actually submitted a work which is under revision, where we showed that these techniques are also applicable with the micro-CT data. So remember the slides that I showed? For example, the accuracy of segmentation was decreasing as I decreased the number of labeled examples. So the blue curve is that curve here, but only showed for a very, very small amount of pixels being labeled. For example, this guy here corresponds to only 3 slices and 45 clicks per slice. So 0.00006% of the data is labeled. You see that the accuracy is quite low. Now, we were able to actually create algorithms, I'm not going to talk about details, it's unpublished, but that can take that accuracy much, much higher and retain it high as we add more slices. Obviously, this is not as good as fully sampled segmentations, but we're working on closing this gap right here. And visually, if the image is here, if I now train, for example, 0.0026% of the data, very small number of labeled examples, while a naive approach creates over-segmentation of the collagen, the proposed approach really is able to capture the essence and properly segment the image. With that, I would like to thank the team, and I would also like to thank the funding agencies that are supporting this work in addition to the SDSC, the PHRT and the CRPP. And if you have any questions, I'd be more than happy to answer.